Hello everyone, in this class we'll learn about muscle spindle. The learning objective of this class would be to understand the importance of muscle spindle and Golgi tendon organ in motor physiology, draw labeled schematic diagram of muscle spindle, describe the function of muscle spindle, understand the role of gamma motor neuron in the control of muscle tone, and appreciate the structure and function of Golgi tendon organ. Muscle spindles are specialized sense organs present in all skeletal muscles in mammals. Muscle spindles are present in large number in muscles that serve fine movements. They are also found in good number in muscles involved in control of posture, especially the muscles rich in slow twitch fibers types. The muscle spindles are named because of their long spindle shape, that is fusiform shape. The fibers of the muscle spindle are known as intrafusal fibers as they are present inside the fusiform capsules of the spindle. This differentiates spindle fibers from extrafusal fibers, regular contractile units of muscle that are present outside the fusiform capsule of the spindle. Extrafusal fibers are regular muscle fibers. Muscle spindles are the receptors that respond to change in the muscle length and the velocity of lengthening. If you see the structure of the spindle, it's about 2 to 12 fibers. There will be 2 to 12 fibers are present in the muscle spindle. Each muscle spindle is enclosed in a connective tissue capsule. The muscle spindle is about 100 micrometers in diameter and 5 to 10 millimeters in length. Muscle spindle lies in the parallel to the regular muscle fibers and their distal ends are attached to the tendon of the muscle or to the sides of extrafusal fibers. This parallel arrangement and special attachment help in the spindle to respond to muscle stretch. Intrafusal fibers are two types, nuclear bag fibers and nuclear chain fibers. So this is the muscle spindle which is showing the inner intrafusal fibers and the outer extrafusal fibers. If you see the intrafusal fibers, in them the nuclear bag fibers are longer than the nuclear chain fibers. These fibers are called as nuclear bag fibers as they are dilated at their center to form bag-like structure and that contain multiple nuclei. Usually two nuclear bag fibers are present in a spindle, the nuclear bag fiber 1 and nuclear bag fiber 2. The bag fiber 1 has low myosin ATPase activity and responds to best in dynamic phase of the muscle stretch, and bag fiber 2 has a high myosin ATPase activity and responds best in the static phase of the muscle stretch. Nuclear chain fibers are thinner and shorter fibers. They are present by the side of the nuclear bag fiber and do not have definitive bag. The nuclei in these fibers are arranged in a row in the form of chain, for which they are called as nuclear chain fibers. Usually 4 or more nuclear chain fibers are present in a spindle. The central non-contractile part of the muscle spindle contains the receptors, whereas the peripheral parts contain contractile elements. The contractile units are absent in the central portion. Thus, stretch of the central part of the spindle activates the muscle spindle. If you see the innervation of the muscle spindles have both afferent, that is sensory, and efferent innervation. Afferent innervation, the afferent fibers for muscle spindle are 1A and 2 fibers. 1A fibers have a larger diameter than the type 2. There are two types of sensory endings in each muscle spindle. So these are primary endings and the secondary endings. Primary endings are the terminal of type 1A afferent fibers. These are also called as annulospiral endings as they are coiled spirally around the center of the intrafusal fibers. Type 1A afferent fibers has two branches. One branch of the 1A fiber terminates on the nuclear bag fiber 1 and the other terminates on the nuclear bag fiber 2, the nuclear chain fibers. Secondary endings are the terminals of type 2 afferent fibers. They're also called as flower spray endings. They appear like flowers. They're usually present on the nuclear chain fibers. Secondary endings are located paracentral part of the spindle. You see the efferent innervation, that is motor fibers. The spindles are innervated by separate set of motor neurons, the gamma motor neurons. They're also called as fusi-motor fibers. These are small nerves arising from the anterior horn cells and are having diameter of 3 to 6 micrometers. They constitute only about 30% of the fibers in the ventral root. That is 70% of the fibers are alpha motor neurons. Because of their size and number, they comprise the small motor nerve system. The gamma motor neurons supply the peripheral parts of the intrafusal fibers. The terminals of the gamma efferent fibers on the nuclear back fiber 1 form plate ending as they end on the motor end plates. And on the nuclear back fiber 2, the chain fibers form trail endings. So this is the structure of the nuclear back fibers, and this is the chain fibers. If you see the innervation, the afferents, the the primary afferent, it is going to innervate both uh, the nuclear back fibers as well as chain fibers. And if you see the secondary afferent fibers, it is going to innervate only the nuclear chain fibers. If you see the uh, efferents, gamma efferents, they are going to innervate both the nuclear back fibers as well as chain fibers, whereas beta afferent, they are going to innervate only the uh, nuclear back fiber. So this is the structure of and their innervation with the intrafusal fibers, that is muscle spindle. The gamma motor neurons are the two types: dynamic gamma motor neurons that terminate on the nuclear back fiber 1 and the static gamma motor neurons that innervate on nuclear back fiber 2 and nuclear chain fiber. Stimulation of dynamic gamma motor neuron increases response in type 1A afferent fibers, that is only during the dynamic phase of the muscle stretch, that is during the change in the muscle length. Stimulation of static gamma motor neuron increases the response in type 2 afferent fibers only during the static phase of the muscle stretch, that is in the maintenance stretch. Thus, gamma motor neuron monitor the muscle length and the speed of the change in length. The muscle spindle is also innervated by beta efferent fibers, and there are both dynamic and stretch beta afferents. The afferent discharge pattern. Two patterns of afferent discharge are obtained following stimulation of the motor neurons, that is dynamic and static patterns. Both uh, gamma and motor neurons produce these two type of responses. Stimulation of dynamic fusimotor fibers results in dynamic response, and stimulation of the static fusimotor fibers produces static response. Fibers from nuclear back fibers show dynamic response, and fibers from the nuclear chain fibers show static response. Coming to the function of muscle spindles are the receptors that are sensitive to stretch. When the muscle spindle is stretched, the sensory endings are distorted and action potential is generated in the afferent fibers. The frequency of afferent signal is proportional to the degree of stretching. Spindle is stretched by the stretch of the muscle as spindle is present in parallel with extrafusal fibers. Therefore, stretch of the muscle initiate activities in the spindle afferents. The spindle afferents directly terminate on the cell bodies of alpha motor neuron in the spinal cord. Therefore, spindle afferent activity stimulates alpha motor neurons that in turn results in muscular contraction or muscle contraction. On the contrary, when muscle contracts, the spindle activity, that is firing in the spindle afferent, is less as the muscle spindle is no more stretched. Thus, the stretch of the muscle causes muscle contraction by stretching the muscle spindle, whereas the contraction of the muscle causes muscle relaxation by inhibiting the spindle. Therefore, muscle spindle and its reflex connection with the motor neuron constitutes feedback device that control muscle length. What is the effect of stimulation of the gamma motor neurons? Muscle also contracts in response to stimulation of gamma motor neurons. But in this situation, the muscle contraction is not direct response to the gamma motor neuron discharge. The stimulation of the gamma motor neuron causes contraction of the peripheral portions that contains the contractile elements of the muscle spindle. This in turn stretches the central portion of the muscle spindle, which contains primary endings. This distorts primary endings in the muscle spindle, which in turn increases the discharge of the afferent fibers. Afferent fibers stimulate alpha motor neurons and cause muscle contraction. Thus, the gamma motor neuron increases the sensitivity of the spindle to stretch. It should thus be noted that the muscle contracts by stimulation of motor neurons directly by stimulation of gamma motor neurons indirectly. In fact, the sensitivity of the muscle spindle to stretch varies with the state of gamma efferent discharge. Therefore, the stimulation of gamma motor neuron is in a stretched muscle leads to maximum spindle response. Okay. So when there will be activation of the gamma motor neurons, it causes contraction of the peripheral parts of the muscle spindle. Okay, as peripheral parts of the contains contractile proteins and they're innervated by gamma motor neurons. Okay, so that causes stretching of the central part of the spindle, and that again distorts primary sensory endings located in the center of the nuclear bag fibers, and that is again going to generate action potential in the 1A fibers, afferent fibers, and stimulation that leads to stimulation of the alpha motor neurons in the spinal cord. Okay, so as 1A fiber directly terminates on the alpha motor neurons in the spinal cord, okay, ultimately that is going to lead to muscle contraction. Okay, you can see the effect of stretch and stimulation of the gamma motor neurons on the muscle spindle activity. So if this is the resting muscle, when stretching of the muscle is going to lead to the stretching of the spindle, and there will be increased afferent discharge activity. And so when there is a gamma motor neuron activity, so there is a contraction of the peripheral parts of the muscle spindle, so that increases the afferent discharge. And you can see the additional effect of muscle stretch and gamma stimulation both can increase the contraction of the when the muscle stretch and the gamma stimulation, they further going to stretch the muscle spindle, and you can see the increased activity in the afferents. Spindle responses. The primary endings on the nuclear bag fibers and nuclear chain fibers are stimulated in response to stretch, but the patterns of responses are different. The dynamic response, the ending on the nuclear fiber region show a dynamic response. That is, they discharge more rapidly when the muscle is being stretched and less in response to sustained stretch. In static response, the primary endings on the nuclear fibers show a static response, that is, they discharge at an increased rate throughout the period of stretch of the muscle. Thus, the primary endings are capable of exhibiting both dynamic and static responses, which means they respond to both change in the length and velocity of the lengthening of the muscle. The static response is the response to muscle stretch, that is change in the muscle length at maintained stretch, whereas the dynamic response is the response to the rate at which the muscle is being stretched, that is the velocity of the lengthening of the muscle. Okay. So here in this diagram you can see that the spindle responses. Okay, so when there is a muscle length changes, you can see in the nuclear back fibers increase in the response. And when the muscle length is reducing, you see the less response in the nuclear back fibers than the normal. But where in nuclear chain fibers, the change in the muscle length uh, will not affect much. Okay. And you can see when the muscle becomes static, there is a constant rate in the discharge in the afferents, the nuclear chain fibers. Control of gamma afferent discharge. Gamma motor neurons do not receive input from primary sensory afferents, rather they are primarily influenced by the descending pathways. The activity in the descending pathway increases the sensitivity of the muscle spindle by adjusting the rate of gamma motor neuron discharge. However, activities originating in the sensory pathways also indirectly influence gamma motor neuron discharge by activating the descending influences. For example, in Jędrysik manure, the process in which the tendon reflexes are better elicited by trying to pull the hands apart the when the flexed fingers are hooked, the rate of gamma motor neuron discharge increases. In Jędrysik manure, proprioceptive input from the hand activates the supraspinal centers, which in turn increases the gamma efferent discharge by stimulating the descending pathways. Tendon reflexes are also exaggerated in anxiety, as anxiety increases the gamma efferent discharge. Alpha and gamma co-linkage. The descending pathways that stimulate the gamma motor neurons also stimulate alpha motor neurons. Therefore, gamma efferent discharge increases along with the increased discharge of alpha motor neurons, and this is called as alpha gamma co-linkage, that is alpha gamma co-activation. Because of this alpha gamma co-interaction, the muscle spindle continues to discharge even when the muscle is in contracted state. Thus, spindle adjusts the motor neuron discharge throughout the period of the muscle contraction. So, this diagram shows how descending pathways are uh, going to affect both alpha motor neuron activity and gamma motor neuron activity. They more so influence on the gamma motor neuron activity. Okay, and when alpha motor neurons are activated, they're also going to activate the gamma motor neuron activity, and that is called as alpha gamma co-activation. Golgi tendon organs. Golgi tendon organs are found in the tendon of the muscles. These Golgi tendon organs are formed by the terminals of group 1B afferent fibers. The diameter of GTO is about 100 millimeters and the length is about 1 millimeter. These sensory endings are arranged in a series with that of muscle fibers. The 1B fibers originating from Golgi tendon organ terminate indirectly on our motor neurons via interneurons. As the interneurons are inhibitory, stimulation of 1B fibers inhibit the motor neuron activity. So these are the sites where these Golgi tendon organs are present, where they're present in the tendon, and they're supplied by afferents 1B fibers. The function of Golgi tendon organ, because of their arrangements in series with the muscle, Golgi tendon organ can be activated either by muscle stretch or by contraction of the muscle. However, strong muscle contraction is a stronger stimulus than passive stretch of the muscle. The passive stretch does not extensively stimulate Golgi tendon organ because the elastic fibers take up much impact of the stretch. But the afferents from Golgi tendon organ discharge actively in response to the contraction as muscle contraction stretches the tendon to greater extent. The actual stimulus for activation of Golgi tendon organ is the force that develops in the tendon, that is the muscle contraction or muscle stretch. Thus, Golgi tendon organ signals the force of contraction, whereas the muscle spindle signals the muscle length. Therefore, The Golgi tendon organ provides force feedback, whereas muscle spindle provides length feedback. Okay. So this is what is going to happen when there is a stretch and some contraction of the uh, Golgi tendon organs. So this is the relaxed muscle. So when the muscle is stretched, then also Golgi tendons are activated. But when there is a strong contraction, uh, so there is a much more activation of the Golgi tendon organs. So this is what uh, is going to happen to the muscle, the force and length feedbacks for the motor control. So when there is a stimulation of the alpha motor neurons, obviously it is going to cause the contraction of the muscle fibers, and the muscle length feedback is given by the usually muscle spindles, okay, to the alpha motor neurons, and also it is also determined by or regulated by the gamma motor neuron activity from the descending pathways, whereas the force of the muscle is going to be detected by the tendon organs, and if it increases, it causes the reflex relaxation of the muscles and reduces the force. So this is in brief about the muscle spindles. Thank you.